My name is Giuseppe Pantaleo. I am a professor of medicine and head of the Service of Immunology Allergy at the Lausanne University Hospital in Lausanne, Switzerland. Today I'm going to present you some recent data that we have generated on the HIV cell reservoir and also on some potential new strategy for targeting this reservoir. Before we go into the actual data, let me give you a brief introduction, a brief background. And then one of the most important questions in the HIV field is to try to understand why does HIV persist in long-term antiretroviral treated infected subject. And as you can see in this cartoon, there are main hypotheses. In particular, I've listed 3 hypotheses, which are not necessarily mutually exclusive, but actually I believe that they, these 3 hypotheses, they contribute to the persistence of HIV. Let's start with the latent HIV cell reservoir. There are a seminal study performed by the Bob Silicano group that have clearly demonstrated many years ago that a population of memory CD4 cells, which in size about 10 to the 5th, 10 to the 7 memory CD4 cells, they are infected with HIV in a latent form. And then what has been estimated is that the half-life of these memory CD4 cells is about 43 months. And then in the presence of complete suppression of virus replication, it will take about 70 years in order to have a physiological elimination of these cells. The problem in getting these cells eliminated is that since the virus is latent, they are not susceptible to antiretroviral therapy, and also they are not susceptible by the immune system because they are not seen by the host immune response. Then there has been another important paper published by Nicolas Chaumont and Rafik Sekali, who has identified in peripheral blood what is the cell population harboring this latent virus. And then what they demonstrated is that the latent virus is predominantly contained in the T central memory and in the T transitional memory CD4 cell population. Let's move now to the residual HIV replication. Now, there are more and more evidence that there is the persistence of transcriptionally active CD4 T cells. These cells may be resistant to the cytopathic effect of HIV. They are poorly accessible to CD8 cytotoxic T cells, and then we will see later why. They may support minimal virus spreading, and also they may potentially replenish the latent cellular reservoir. There are a series of studies that recently support the presence of residual HIV replication even in the presence of antiretroviral therapy. A recent paper published by the Walensky group in Nature. There is another interesting paper published by Timothy Shaker and his colleagues, where it has been shown that even in patients that are treated as much as 15 years with antiretroviral therapy, when it is stopped, the virus will come back, will rebound, with a mean average of days around 17 days. Again, suggesting this rapid rebound that when RT is stopped, there is already ongoing virus replication responsible for this virus rebound. There is another recent paper published by John Mellor's group where they are showing that indeed for a long time after ART, there is still a decay of the plasma viremia. Then in individuals that have plasma viremia below 50 copies, it's going to take many years, up to 8-10 years, in order to have this patient to become completely undetectable. Finally, there is also another study from Timothy Shaker that he was suggesting that maybe the antiretrovirals in the tissue do not reach sufficient concentration to make sure that the virus is fully suppressed. Finally, the last hypothesis is the presence of privileged anatomic compartments and the presence in this compartment of massive T-cell activation. And then here I'm showing a paper that we published about now 23 years ago. And then I want to make 3 points. First of all, on the top left panel, there is a germinal center. There is a section of a lymph node with many germinal centers. It's very important to keep in mind that these germinal centers, they are the area where the immune response is generated, particularly the antibody response. And then you can see in the bottom panel, you have these white areas that correspond exactly to the germinal centers that are positive for in-site hybridization for HIV RNA. Then these germinal centers in viremic patients are packed potentially with HIV virus particles. And then in the panel on just on the right, in the middle, it's a staining for CD21, which stains follicular dendritic cells. And then there is in-site hybridization with positive HIV RNA. There are these black dots. And then again this indicates that the HIV virus particle will be present on the extracellular surface of these follicular dendritic cells. Now, then a few years ago also, we, as follow-up of this study, we continue over many years to study lymphoid tissue, and then we published a paper that was drawing the attention on the importance of T follicular helper cells, which is a population of memory CD4 T cells, which is exclusively present within the germinal center. And then what we showed that in viremic patients, these cells serve as the major CD4 T cell compartment for HIV infection, replication, and production. And then the main result from this study was that when we tried to isolate virus from lymph node cells from this patient, we were able to isolate virus only from cells expressing CXCR5 and PD-1, that are the two markers defining T follicular helper cells, and also from cells expressing PD-1, whereas was hardly possible to isolate from other cell population. Then the other interesting observation was that we look at the correlation between the percentage of these T follicular helper cells and the levels of plasma viremia, and then we found a nice correlation. Then what we did also is to sequence the virus from these different cell populations in the lymph nodes, and then we compared to the virus that was present in the plasma. Then what we found that the virus in several patients that found in the plasma is identical to those found in this population from the lymph nodes. Now, again, the importance of the germinal center. Why we think that the germinal center are the primary site for production and persistence of HIV? The germinal center are a very important unit from an immunological standpoint, and when there is an ongoing immune response, the presence of germinal center reflects an activation state of the lymphoid tissue. And then in an active germinal center, we have two areas. One where there is massive division of B cells, which is called the dark zone. And then when these T cells, they go through maturation, they will leave the dark zone to go in what is called the light zone, and to remit the antigen that is expressed on the extracellular surface of follicular dendritic cells. And then in order to be further selected, they will receive the help from the T follicular helper cells. Now, why these germinal centers are so important also for HIV infection? Well, first of all, because as I have shown you just a few slides before, on the extracellular surface of these follicular dendritic cells, there are the virus particles are trapped. And then they may represent a continuous source of virus for infection. Second is the exclusive place from an anatomic standpoint where the T follicular helper cells are present. And then we can imagine that the T follicular helper cells, when they enter the germinal center, they can get either infected from the virus that is trapped in the FDC network, or if these cells are already infected but in a latent form, when they enter the germinal center, they will be reactivated and then to start to produce virus again. Then it's also known that generally cytotoxic CD8 T cells are not very numerous in the germinal centers, and then there is the potential imbalance between the ratio between effector CD8 T cells and the target cells that need to be killed. And then finally, in the germinal center, there are unique conditions for HIV production because of the massive T cell activation of TFH and also of the presence of cytokines that will support HIV production. Therefore, the germinal center, they function as HIV production units. Then I will go over a number of issues in the next 10-15 minutes. First of all, the first 2 points, we will address the cells that are serving as primary cell reservoir for replication competent and infectious virus in a long-term treated RT subject. We also will investigate what is the compartment that is supporting residual HIV replication in long-term treated subject. And then we will look at new strategies to target this repertoire with antibody therapy and the effectiveness of this therapy to eliminate this repertoire. Now, in order to perform this study, we have enrolled 18 patients that are all long-term treated between 1 to up to 15 years, all viremia below 50 copies. And then we performed either viral growth assay or cell-associated HIV RNA. And then we collected simultaneously blood and lymph node biopsy from this patient. Then before going into the virologic data, this slide is showing what are the changes in the different cell population occurring in the lymph nodes and the difference between lymph nodes and blood from healthy subject, from HIV viremic and HIV treated. The only thing that I want to point out is that the percentage of T follicular helper cells is dramatically reduced after long-term therapy. Then, and the other thing that I want also to underscore here, where we have investigated some activation markers such as HLA-DR, CD38, Ki67, you can clearly see that in the viremic individuals, these markers are highly expressed in T follicular helper cells, but not in the other cell population, which explains why this TFH will support better virus production. And then antiretroviral therapy decrease this activation, but not sufficient to completely bring down to normal level that we observed in healthy individuals. Now, because we had, the percentage of TFH is so low, then we could not perform individual sorting of the different cell population, but what we decided to sort total PD-1 from blood and lymph node and then double negative as single CXCR5 positive cells. And then just to underscore that about 65% of the cells that are total PD-1, they contain, are represented by TFH cells, then they express CXCR5 and PD-1. And then what we did were virus outgrowth assay. Then in order to determine the presence of replication competent virus, and then what is the classical virus outgrowth assay? Then we take the cells from a patient, we mixed the cells with CD8 depleted cells from HIV negative, we stimulate with anti-CD3, anti-CD28, we follow for 14 days, and then at day 5, day 14, we measure the presence of the virus through HIV RNA and P24. Then something that I want to underscore is that here we are using 100,000 sorted cells as compared to what is generally done using blood, where people use generally 2.5 million, 3 million cells per well. Then from the lymph node, we use 25, 30-fold less cells to perform this assay. These are the results. You can see that we can isolate HIV RNA from blood and from lymph node, higher level from the lymph node. But when we look at P24, we could isolate P24 predominantly, almost exclusively, from lymph node PD-1 positive cells, but not from the blood. And then you can see here the cumulative data clearly indicating that at day 14, there are as much 70,000 more HIV RNA in the wells from PD-1 cells from lymph node as compared to the other cell population. And then with regard to P24, as I said, we found almost exclusively in the culture from PD-1 positive cells. Interestingly, there was a correlation between the levels of HIV RNA produced and the level of P24, and we identified a threshold at 10 to the 4th in order to detect P24 in the culture. Something here very interesting is also you concentrate predominantly to the bottom panels. You can clearly see that when we look at the percentage of TFH and then the levels of HIV RNA of P24, there was a nice correlation, clearly indicating that these cells were mostly responsible for the production of HIV RNA and also of P24. Then the other question was to try to understand whether these cells were producing infectious virus. Then what we did, then we took cells, CD8 depleted from HIV negative subject. We stimulated for anti-CD3, CD28, and then we exposed these cells to the virus of the growth assay supernatant for 6 hours. We washed, and then we followed for 14 hours, and we measured HIV RNA. And then as you can see here, then 60% of the subject were positive from the culture for PD-1 positive cells. And then we isolated then infectious virus predominantly from lymph node cells and predominantly from PD-1 positive cells, but not from blood. Then what we found also that there was a negative correlation between the levels of production of HIV RNA and duration of treatment, then indicating that treatment is reducing the levels over time. What we found is the same also for P24, but we found detectable P24 only for the first 3 years after the initiation of therapy. Then the first conclusion that we can make is that the results of the VOA demonstrate that TFH cells are the major source of inducible HIV-1 in our treated subject. PD-1 positive TFH cells in blood and lymph nodes are the greater producer of HIV replication competent infectious virus, but these observations do not inform us of whether these cells are responsible for the persistence of HIV replication or production in long-term treated. Then in order to better understand the role of these cells in supporting the persistence of HIV replication or production in this long-term treated subject, we assessed the presence of cell-associated HIV RNA in the different memory CD40 cell population. And then as you can see in this data, again, these PD-1 positive cells were the cells that had the higher level of expression of cell-associated HIV RNA as compared to the other. And interestingly, when we look at the changes with the duration of the treatment, what we could show is that there was a decline over time, but we could find the detectable cell-associated HIV RNA up to 12 years after the initiation of treatment. And more interesting, we did not see much decline for the first 4 years after initiation of treatment, but only after the first 8 years. Therefore, the conclusion for these data indicate that these cells have the highest level of cell-associated HIV RNA, thus indicating that this cell population is the primary site for virus replication and or production. This cell-associated HIV RNA was detected in patients treated from up to 12 years. That demonstrated these cells are responsible for persistent virus replication or production. A negative correlation was observed between the levels of cell-associated HIV RNA in this cell population and duration of treatment. and levels of cell-associated HVRNA were stable for at least 5 years after ART, and the initial decline was observed in subjects treated for at least 8 years. The lack of decline in the level of cell-associated HVRNA in PD-1 positive TFH cells for up to 8 years, and their persistence up to 12 years, may help to explain why virus rebound is consistently observed after ART interruption. Now, another important question is to look at the changes occurring in the lymph node tissues after long-term ART. And then, as you can see in this lymph node section, this is a lymph node biopsy obtained from a patient treated only for 3 months. You can clearly see a lot of germinal center that are easily, visible. And then you can clearly see here, where you see these cells that are stained within the germinal center are PD-1 CD4 T cells. Here is at higher magnification, and these correspond to T follicular helper cells. Now, in a lymph node section from a patient treated for 10 years, you can barely recognize germinal center at this lower magnification, but at higher magnification, we can recognize a couple of small germinal center, again with PD-1 positive CD4 T cells. Whereas in a patient treated for 12.2 years, we cannot recognize any more germinal center, but we can recognize only a primary follicle with very rare PD-1 positive cells. And then interestingly, what we found is a negative correlation between the number of germinal center and the duration of treatment, as well as the number of PD-1 positive CD4 T cells and the duration of treatment, clearly indicating that there is a shift of activation of the tissue over time with this loss of germinal center following antiretroviral therapy. Therefore, long-term ART was associated with a progressive shift from an activation to a quiescent state of the lymph node tissue, as indicated by the progressive reduction in the number of germinal center, and was also associated with a progressive reduction in the number of PD-1 positive TFH cells. Then the final question for the cell reservoir, we wanted to understand what was the frequency. Then we look at the frequency, evaluating quantifying the number of RUPM and IUPM. Then we quantify the number of CD4 T cells per million of CD4 T cells containing replication competent of infectious virus. And you can see here that we have quantified that there are about 37 cells in the PD-1 population per million CD4 T cells containing replication competent virus, and 6 cells containing infectious virus. Now, even if we adjust for the different representation of the different cell compartment within the CD4 T cell population, what we can show that still the PD-1 positive, that is a minority population, accounts for 46% of the cells containing replication competent virus, and more importantly, for 96% of the cells containing infectious virus. Therefore, the PD-1 positive TFH cell population is the major cell compartment of cells containing replication competent and infectious virus. Now let's move to the last 5 minutes to looking at the targeting with the antibody therapy and the efficiency. And then here in this cartoon, you can see different strategies that are being evaluated to target the reservoir. There are a recent study that has been using what is called the BITE, which is a strategy that allows to activate CD8 T cells. And then what we have been working is on another strategy, and based on this data that we think that the major target is PD-1, to use anti-PD-1 antibody coupled with toxins. I'll show you data only on the anti-PD-1 antibody coupled with toxin. Now, here what we have used is a toxin, and then the only thing that I want to say about this toxin is that it has the peculiarity that it kills both resting and dividing cells, and then most of the other toxins used in the cancer field, they only kill dividing cells. In our case, it was important to have a toxin killing both dividing and resting cells. Now, here are the data, and then here showing only the efficiency of this antibody to kill these PD-1 high positive cells. Here on the left, you have a PD-1 low of intermediate PD-1 positive cells. You can see that this in red is the antibody anti-PD-1 that is not coupled to toxin, in yellow coupled to toxin. Then here is another antibody not coupled to toxin, coupled to toxin. As you can see, there is not much cell killing, there is not a specific killing. Then if you go on the PD-1 high, you see the uncoupled PD-1 antibody that, but when it's coupled with toxin, you can see that there is from about 35 to almost 80% of annexin positive cells, which is a marker of apoptosis. And the same, you see a shift in killing with the other antibodies, with the coupled toxin antibody. Then we get on the annexin positive, we look of aqua positive cells, which is a marker of cell death. And again, you can see from the red to the yellow, you see a big shift. Then we have about 60% of the cells that are annexin positive, that are dead. And then with the other antibodies, about 45%. Then these two antibodies, coupled with toxin, were very efficient in killing PD-1 high cells. And then in this experiment, we look also at the changes after treatment with the integrated HIV DNA. Then you can see that in the cell population treated with the antibody plus toxin, we can see a decrease of about 0.6 logs in the frequency of cells containing integrated DNA. But the most important question was to understand whether we could prevent suppression of, we could prevent isolation of virus and infection in vitro. Then what we did, we took then again the cells from a patient, okay? And then we have a control cells that are not treated with the antibody. Then we have a control that of the positive control, the cells that are treated with an antibody coupled with the toxin. And then for 5 days, then we check the viability at day 5. And then what we do, we perform a quantitative viral growth assay for 14 days, adding CD8 depleted allogeneic cells from HIV negative. Then we stimulate with anti-CD3, anti-CD8 MABs. And then we measure HIV RNA at P24 at day 14 and at day 5. And these are the data. As you can see on the top, you can see the killing that we can get very nicely in PD-1 high between the antibody uncoupled to toxin with those that is coupled. Again, about 80% of cells are annexin positive, about 60% of the annexin positive are aqua positive. And then if you look at the bottom, here you see at the RUPM or the IUPM, and then you can see this is the control that has not been treated with the antibody. Here is the positive control that is stimulated with anti-CD3, CD28, the anti-CD3, CD28 plus the isotope control for our antibody anti-PD-1 that is boosting the virus isolation and replication. And then you can see here in white, there is full suppression of the isolation of virus when we, in the culture that have been pre-treated with anti-PD-1 antibody coupled with toxin. And then it's exactly the same on the right side when we look at the presence, the isolation of infectious virus, complete suppression. We repeated the experiment using 2 antibodies, and then the top is giving you the frequency of cells that are lysed, and then the bottom again are showing you this is our positive control, the antibody uncoupled, the antibody coupled, full suppression, uncoupled antibody is a different anti-PD-1 antibody, coupled full suppression, and the same for the isolation of infectious virus. Therefore, if we conclude with a couple of cartoon slides, just to recapitulate, we think that the Germinal Center represents really the HIV production unit. This is the situation in a viremic patient where there is the Germinal Center are loaded of virus particles and infected cells, and then they will be mostly responsible for the production of viremia. in an individual that is treated with, long-term treated with ART, still there is the possibility through the TFH that there is reactivation of virus from cells that were latently infected, that when they enter the germinal center, they will become massively activated, or they can replenish the reservoir through the production of virus particles that can be trapped in the extracellular surface of oligodendritic cells. And these reactivation can be responsible for the transient blips that we observe in long-treated patients. Now, following treatment also, there is a progressive shift of the tissue from an activation to a quiescent stage with the loss of germinal center, and then also there is a progressive loss of PD-1 positive cells that correspond predominantly to TFH. Therefore, the final conclusion is, taken all together, these results indicate that despite the lack of detection of viremia, active and persistent virus replication or production occurs in the lymph nodes many years after ART. The persistence of active virus replication is likely driven by the state of activation within the lymph node tissue, and at least 10 years of therapy are needed to induce a quiescent state in the lymph node tissue and a substantial reduction in the pool of cells supporting active and persistent HIV replication during ART. Therefore, 10 to 15 years of ART may be needed to effectively suppress persistent virus replication in lymph nodes. Finally, let me thank my collaborators, in particular two students, Ridima Bagga for the cell reservoir study, Alexander Rockinger for the antibody study, and the two senior scientists that have led with me this project, Mathieu Perrault and Craig Fenwick. Thank you.